Good afternoon and welcome to the CHAP Phenotyping Laboratory webinar. I hope this is where you're meant to be, and you're very, very welcome. First, a few IT housekeeping issues. All videos and microphones will be off during the webinar, but please feel free to type in any questions into the Q&A box. And if you have any problems, use the chat function. As you heard just now, the webinar will be recorded and we can send you the link afterwards. So moving on then to speakers and the agenda. We have this motley crew. We have Dr. Tom Ashfield talking about the CHAP lab based at Rothamsted. Then we have, and Tom is a scientific specialist at Rothamsted. Then we have Tom Walters, who is the head of intelligence at the small robot company, and he's talking about a case study that they've done. Then we have Charles Vase, who's the managing director of Fotenix, talking about another webinar that we have done. And finally, we have Michael Carstensen, who is CEO of the Videometer, which is a company that's supplying us with the new seed sorter that's coming soon. And then we'll have a Q&A. But first of all, just a little bit about CHAP. For those of you who don't know about us currently, we're one of 4 centres funded by Innovate UK and established in about 2014 to 2016. There are 4 centres. There is CHAP, which looks at crop health and protection. There is AgriEpi, looking at engineering and robotics. There is CL, who look at Centre for Excellence in Livestock, and there's Agrimetrics, who look at big data. And CHAP's vision for the UK is to be the global leader in the development of applied agri-technologies and to secure our future by nourishing a growing population sustainably while delivering economic, environmental and health benefits to society. We do that by working with our partners to provide open access to our advanced facilities run by world-class experts. And we invest and manage commercial demonstration facilities that field test and showcase the benefits of new technologies to accelerate their adoption. The true benefit, as you'll see from Rothamsted, is that we're based there with their expertise as well. So we have our own lab, we have Tom, and we also have the other scientists of Rothamsted and their facilities, which is really great for potential collaboration and for customer projects. So the purpose of CHAP is to accelerate the identification and development and adoption of agri-tech solutions that deliver greater sustainability for growers and populations. And we connect industry with the best researchers, innovators and government resources. We're independent and not for profit. This is an overview of our partners. We can see we're scattered all over the UK currently. We have the 4 non-delivery partners, Bayer, ADAS, Frontier and Tesco. And then our delivery partners are the partners where we have capabilities based. So we have Liberty with JHI up in Dundee. We have RIPE, who are based with Warwick University. We have Cranfield, where we partner with our AgriEpi centre with a large phenotyping gantry. We have STC, FERA, AHDB, obviously Rothamsted, where we're focusing today. We have CABI down in Surrey, Newcastle University, and the latest partner is NIAB. And then at each of these partners, we have these various capabilities. So we have the eFlow mesocosm with FERA. We have Crop Monitor Pro, again with FERA. We have a biopesticide development lab with CABI and a fine phenotyping lab at Rothamsted, which we're focusing on today. We have our national reference collection. We have field-based labs. We have a plant clinic. We have molecular diagnostic lab. We have a resistance diagnostic lab. We have international pest horizon scanning, and we have strip-till at STC, crop research sprayer at JHI, crop storage at HDB, soil health facility at Cranfield, and the Advanced Glasshouse Vertical Farming Centre, ICEA, which is the Innovation Hub for Controlled Environment Agriculture, and a Natural Light Growing Centre. So there's a whole vast range of different capabilities that we can talk to you about maybe on another day. But anyway, today we're here to talk about the phenotyping at Rothamsted, so I'll pass you, no, I won't, one more slide, sorry. I just wanted to also talk to you about our membership. You can see here we've got a range of members to CHAP, academia, large companies, SMEs, and so this gives you an opportunity to network, to get bespoke support, collaboration work, workshops, grant opportunities. So if you're interested in becoming a member, then I'll leave my email up at the end and just contact me and I can arrange that for you. I will now hand over to Tom, to his capable hands, to introduce you to the CHAP Phenotyping Lab. Thank you.
Hello, so my name is Tom Ashfield and I'm responsible for overseeing the science in the CHAP Fine Phenotyping Lab. So this facility was set up as a partnership between Rothamsted Research and CHAP with funding from Innovate UK. And in essence, what we do is use some very nice imaging equipment to automate the quantification and remote detection of plant traits and responses. One particular focus of the lab is on biotic threats, so pathogens, pests and weeds. We're also very happy to look at other traits of interest. So the facility itself is located at the Rothamsted Harpington site, and this brings at least two advantages for the services we can offer. So first of all, it means we have access to the enormous depth of crop-related expertise located within the Institute. And this can be brought into projects, either when Rothamsted's a project partner or through consultancy arrangements where individual Rothamsted scientists agree to participate in a project and provide access to their expertise and sometimes resources. Then the second advantage I'd highlight for being located at Rothamsted are the really fantastic facilities located on site. And I just illustrate that with the plant growth facilities here. So we have extensive glasshouse space, we have polytunnels, sand beds for growing plants under ambient conditions. And all these can be costed and brought into projects as required. So again, it's an enormous asset for what we can offer. Okay, so before I go any further, I should just describe what is phenotyping and how can imaging help. So in essence, phenotyping is nothing more than scoring or quantifying plant traits and responses. And sometimes these may be fairly straightforward traits like plant height or leaf number. Other times they may be more slightly harder to score, like for example, resistance to a disease or photosynthetic efficiency. And obviously phenotyping is enormously important to many aspects of crop science. So for example, if you're a plant breeder, you obviously need to score a lot of different traits in large populations. While if you are developing a biocontrol agent, again, you're going to need to assess its efficacy, so you'll need to conduct the appropriate trials and score the appropriate responses. Okay, so that's phenotyping. So how can imaging help? So I'd highlight 4 advantages for image-based phenotyping. So firstly, it can remove some of the subjectivity that's associated with manual scoring. And this is going to be especially true for traits that are not plus minus. And I illustrate this here with a sample of septoria symptoms on wheat leaves on the top row. So I think you'd agree that if you're 2 investigators trying to score those symptoms, there's a good chance they come up with different scores. However, with image-based phenotyping, as I've illustrated with that multispectral image on the right, the same analysis pipeline is always going to come up with the same score for the same sample. So again, that reproducibility is really important. Okay, then the second advantage I'd highlight is you can score things you can't see with the naked eye. An example of that would be photosynthetic parameters. So again, you can't see them with the naked eye, but there are sensors that allow you to measure them. Okay, then a third advantage I'd highlight would be the potential for developing high throughput phenotyping assays and pipelines. And this is possible because you can automate both the image capture and the image analysis. And I just illustrate this with a sample of leaf discs. So again, this is a leaf disc feeding assay. And in the middle, you can see the original images. And then on the right, you can see the images after the image processing to quantify the insect damage. Okay, and then the final advantage I'd highlight is that the image-based phenotyping greatly facilitates data capture at multiple time points during a growth trial. So if you're scoring a trial manually, you're probably going to be limited. The number of times you can score that trial for traits is quite possibly going to be limited by the amount of labor you have available. However, if you're using image-based phenotyping and have brought in automation, then it's sometimes possible to actually capture to score the traits as often as you want. So I just illustrate this with a very nice video provided by Robotics Systems, from whom we've recently purchased some chambers. So just get this running for you. So these chambers have an integrated RGB camera, and here you can see tomato plants just growing and then being imaged every hour, at least during the light period. So in this case, it's just a demonstration and the plants are all healthy, but I think you can see the potential for this type of system to compare the growth of plants, under different growth regimes with different products, and then to be able to capture data and score the traits as often as you want. Okay, so what specific capabilities does the Fine Phenotyping Lab have, and what project areas are we active in? So as for the capabilities, I'll divide these into two general areas. So first of all, there's the imaging equipment. And on the top image, you can see on the right, the LemonTech PhenoCenter. So this is a bit of kit for automated imaging. And then on the left, on the benchtop, you can see like a dome-shaped instrument. So that's the Videometer lab. That's one of our multi-spectral bits of kit. And in the next few slides, I'll actually be talking about those two bits of equipment in slightly more detail and some of the specific applications and uses. And then the second sort of equipment that second type of equipment I'd like to highlight are our controlled environment chambers. And actually, these are really important to what we do, because a lot of the projects we're involved in involve us, first of all, generating the experimental material with the traits that our partners are interested in. So whether this be abiotic stress, pathogen infection, or pest infestation. And again, often we might be working with a Rothamsted scientist with expertise for that particular trait. And then obviously once the material's been generated, we can then image it with the sensors in the lab as appropriate. In terms of the project areas, so just highlight two areas where we're active. So first of all, quantifying plant traits or plant responses under laboratory conditions. So again, this might include, for example, assessing product efficacy or assessing germplasm potentially. And then down the second point, or the second area to highlight, would be where we work with partners to develop remote sensing applications for sensing traits either in the field or the glasshouse. And for example, this might involve detection of pests, pathogens, or weeds. And as you'll hear, I think from the other speakers later in this webinar, often our role in this type of project is to generate the experimental material and do the imaging, and then we'll have other project partners, say, developing the sensor or the robotics. Okay, so now I'd like to just highlight those two bits of kit that I previously mentioned. So first of all, the LemonTech PhenoCenter. So this is a bit of equipment for automated imaging. You can either load the samples manually, you can also load them robotically, at least depending on the sample type. Then the image capture and the image analysis is automated. We have various sensors installed in the equipment. And these include a standard RGB or color camera, together with more sophisticated cameras. So for example, a PAM photometer for measuring photosynthetic parameters and a 3D laser scanner. In terms of applications, again, I highlight two areas. So again, first of all, sort of lab-based trials where products are being assessed under carefully controlled conditions. So I think what the type of project I'm talking about here is, for example, if you've got different herbicide formulations, you want to compare their efficacy. Potentially, if you had different fertilizer formulations or biostimulants, you wanted maybe to assess how they perform in, say, mitigating, I don't know, abiotic stress. So that sort of trial. And then the second general area, or the second application area I'd highlight, would be quantifying pathogen symptoms and pest damage. Okay, so I'm now going to just, on the next two slides, just expand on those 2 points in slightly more detail. Okay, so first application area, lab-based trials to assess product efficacy. And again, the things here we could be looking at, as I mentioned before, herbicides, biostimulants, pesticides, that type of product. And I think the advantage of using the PhenoCenter over manual scoring really comes back to the points I made earlier. So again, it can remove some of the subjectivity associated with manual scoring. You can obviously now image with multiple different sensors here, so you can capture data you couldn't actually capture using the naked eye. And there's also potential, depending on the sample, for also trying to make a more high throughput assay. So just an example, I show here a sample of nutrient stress rocket imaged with some of the sensors, just to give you an idea of the output and some of the sort of traits you can extract. So on the top left-hand corner, you can see the sample imaged with the standard color camera, the RGB camera. So again, for this type of trial, you could look at, for example, chlorosis. If you were looking at a trial involving, say, disease resistance, then you could be scoring something like a lesion size. So essentially anything that's color-based. Then on the top left-hand corner, you can see the image taken with the fluorescence camera. And again, for this type of sample, good for calculating the projected leaf area. So you can see how that changes over time. Down the bottom left, again, the image taken with the panthometer. So this is an instrument that measures photosynthetic parameters. So again, for this type of trial, it can detect chronic stress. So again, you can potentially look how your product can mitigate that. Also very useful for looking at early herbicide responses. And then down the bottom right, that's now the output from the 3D laser scanner. Again, for this type of sample, the potential there is to look at the canopy height and how that's changing as the sample grows, as the trial progresses. Okay, so then on to the second application area that I wanted to mention, so quantifying pathogen symptoms and pest damage. Just illustrate this on the left with quantifying insect damage in a leaf disc assay, and on the right with septoria symptoms. And I think the important additional point I'd like to make with the image on the right is that while it'll often make sense to capture the images using the Videometer cameras, in some cases that's not going to be possible. So for example, in the case of the septoria image or septoria sample, that involved leaves laid out on a metal plate that couldn't be conveniently imaged in the Videometer. So in that case, it's perfectly acceptable to image the sample elsewhere, as long as the lighting, et cetera, is consistent, and then we can analyze the images later. So for this type of sort of quantifying pathogen or pest damage or infestation, so the applications here would include things like assessing or comparing resistance in germplasm, or comparing the efficacy of different, say, fungicide formulations. Okay, so then moving on to the second type of equipment that I'd like to highlight, or the second equipment capability I'd like to highlight. So this is our multispectral imaging capabilities. So we have both lab-based and portable systems. In terms of the lab-based systems, on the left, you can see the Videometer lab. On the right, you can see the Fotenix Delta system. And you'll be hearing a lot more about that and its specific capabilities from Charles Vase, who'll be speaking later in the webinar. In the middle, you can see the Videometer camera. So that's one of our portable cameras suitable for imaging in the glass house or field. For those of you not familiar with this type of camera, for multispectral cameras, essentially what they do is they capture images with a range of different wavelengths. And in the case of our lab-based systems, these range right from the UV through the visual range and then into the near infrared. And then from these images, you can calculate reflectancies and then essentially plot these out as sort of spectral signatures for the particular trait of interest. And why multispectral imaging can be really useful, and its advantage over standard RGB photography, is it sometimes allows you to distinguish really subtle traits that would be hard to actually quantify in a standard color photograph. And I just illustrate that on the next couple of slides. Okay, so in terms of then the applications, so first of all, again, multispectral equipment can be really good for lab-based phenotyping, illustrated with two different samples here. So the first would be for quantifying root disease. And what you can see here is actually takeover disease on wheat roots. On the left, you can see the sort of image shown as a color image. And I think you'd agree that it's quite hard to actually see the symptoms and to quantify them in that image. But on the right, you can now see a false color image based on analysis of the multispectral data. And in this case, the disease symptoms are shown in blue. So again, we're getting robust detection of the symptoms, and obviously that then gives you potential to compare germplasm or biocontrol agents or whatever product you're interested in. And again, I should mention this was developed together with the Rothamsted Research group. Down the bottom, the second example, so this is now detecting fusarium growth on grain. And it's a similar story. So on the left, you can see the color photograph, So again, I think it's quite hard to actually see the growth on those grains in the color image, and it'd be quite hard to train a computer pathway to actually to quantify that. But in the force color image on the right, so again, that's based on the multispectral data, that we're getting robust detection of the fusarium, and you can see the contaminated grains shown in red. And we're really excited actually about this capability of using multispectral imaging to detect seed and grain related traits, because later this year we're acquiring a robotic seed sorter from Videometer that will allow us to sort seed based on just this type of trait. So I think that's going to be a really useful addition to our capabilities. And you're going to be hearing more about that from Michael from Videometer, who will be speaking later in this webinar. Okay, and then the second area of application for multispectral imaging I'd like to highlight would be for developing remote sensing solutions in the field of glasshouse. We've been involved in a number of projects of this type, and here I just highlight two. On the top, the fruit condition project. So again, this focused on developing the capability for the sort of remote quantification of fruit ripeness and condition in soft fruits, specifically tomatoes and strawberries. So, and again, the partners in this project were Rothamsted Research and Fotenix. And on the bottom, I highlight the second project. So this is the SlugBot project. It's an ongoing project and focuses on remote detection of slugs and the precision application of treatment. And in addition to CHAP, the partners in this project are the Small Robot Company and Lee Farmers. I'm not going to talk more about these projects because both Charles from Fotenix and Tom from Small Robot Company are going to be talking after me. So I think you'll be hearing more about this type of sort of remote sensing project from them. So what I'd like to do is just finish up now with really summarizing four areas where I hope we could help you. So first of all, could work with you to assess product efficacy under carefully controlled conditions. Secondly, to assess whether imaging is suitable for your particular phenotyping or remote sensing application. So essentially, we can do some of that initial work with you just to determine whether image-based phenotyping is going to be suitable for what you want to do before you actually have to invest in some potentially quite expensive equipment. Thirdly, we can work with partners to develop remote sensing solutions, as you saw on the previous slide. And then finally, we can also provide a test site for new sensors. So we can generate the experimental material traits that you're interested in, do the imaging potentially, and then help you determine whether or not your equipment is going to be suitable for that, and potentially how it needs to be modified or developed further. Okay, so with that, I will finish up with my contact details, and I hope you enjoy the rest of the webinar, and I'll be very happy to take questions at the end.
Thank you very much, Tom. As Tom said, we'll take questions at the end, but if you think of anything as we're going along, please feel free to bung them in the Q&A tab at the bottom of the screen, and then we'll review them when we're finished. So now I'd like to hand on over to second Tom from Small Robots, with a very impressive job title. I hope you enjoy this one as well.
My name's Tom Walters, and I'm Head of Intelligence at the Small Robot Company. So the Small Robot Company is building systems that enable per-plant farming. We survey fields in fine detail with a robot, and then we use the information that we gather through those surveys to provide detailed feedback to farmers to allow them to make decisions, and ultimately to enable our robots to treat plants on an individual basis. This opens up applications like non-chemical weeding and the precision treatment of individual plants in a field. Now, to realise our vision, we need to be able to accurately identify the crop plants, the weeds, and the pests in a field. And by partnering with CHAP's Phenotyping Lab and their state-of-the-art capabilities in this area, we've been able to significantly increase what we can do with these robots. And so they're a natural partner for our research and development efforts. So to give you a flavour of what the kind of thing that we do, this is Tom. This is our field survey robot. It has a boom with 6 cameras mounted on it, which take high-resolution images of the field as the robot drives along. Using the system, we can quickly gather hundreds of gigabytes of image data from a field, and all of this information is then fed back to our centralized system known as Wilma, which analyzes these images to detect the individual plants growing in a field. In this slide, you can see our existing broadleafed weed detection system in action. On the right is an image from a survey done by Tom, and we've trained a machine learning algorithm that can detect the locations of the individual broadleafed weeds in this image. We can then take those individual detections and display them on a map of the field, dividing into cells, showing the overall density of the weeds in each area. This map is then made available to the farmer, who can then make use of this information to make decisions about when and how to treat the weeds. So using our system, the farmer can choose to make decisions as to how to treat very small regions of their field down to the level of individual plants, rather than having to make decisions at the level of the whole field. So from this example, I think it's clear that accurate identification of individual plants is key to the success of our systems, and this is an important aspect of our continued research efforts. So to that end, we have a couple of ongoing collaborative projects with the Fine Phenotyping Lab at CHAP. We have one around blackgrass detection. Now, blackgrass poses an interesting challenge because it's a weed which grows with wheat. Being a grass, it looks almost identical to wheat at the early stages of its growth, which is when you want to treat it. Using hyperspectral imaging, we've been developing systems that can detect blackgrass within wheat crops when the plants look almost identical to the naked eye. The other project I want to talk about is the beautifully named SlugBot, which is an end-to-end robotic system to eliminate slugs by precisely spraying the individual slugs with nematodes, which are a slug parasite, rather than using harmful chemicals. Both of these projects unlock key new skills for our per-plant farming platform by expanding the range of things that we can identify and act on. So per-plant farming allows for the targeting of pernicious weeds when they emerge, while leaving beneficial non-crop plants in place. Blackgrass is definitely a pernicious weed. It's been estimated that the total cost to UK farming from blackgrass is about £400 million each year in lost productivity. And blackgrass detection, as I mentioned, is particularly challenging because blackgrass and the crop plants are morphologically very similar, especially at the early growth stages. If we can crack the problem of detecting blackgrass at these early growth stages, then it makes treatment and removal possible, potentially using the electric weeding systems that the small robot company is currently developing. So this collaborative project draws together expertise from the Fine Phenotyping Lab at CHAP, the Weeds Group at Rothamsted, the Small Robot Company, and Fotenix with their expertise in multispectral imaging. And you'll be hearing more from their CEO, Charles, in the session following mine. And the goal of the project is to detect blackgrass using multispectral imaging and ultimately to build a system which can do this in the field. Phase 1 of this project involved lab trials with CHAP's advanced hyperspectral imaging facilities, along with expertise and growth facilities from Rothamsted. The data from these initial experiments were analysed by Fotenix and showed some promising results on being able to differentiate blackgrass and wheat at the early stages of their growth. Phase 2 of the project is ongoing and involves in-field multispectral imaging using a combination of robot hardware developed by the Small Robot Company and multispectral imaging hardware developed by Fotenix. This study is ongoing, but the initial results are looking promising, and we believe that we will be able to scale this up. So I think this is a great example of an end-to-end project that's gone all the way from the lab to the field and has been facilitated by CHAP's capabilities, augmented by expertise from Rothamsted in the form of their Weeds Group, especially David Comont and Richard Hull, who are able to grow and provide the plants that were then imaged by CHAP. The initial hyperspectral imaging studies at CHAP then enabled further research by Fotenix and the Small Robot Company to take those results and build a working multispectral prototype for use in the field. SlugBot is all about the identification and localization of slugs in images and then being able to get spray to them. This system is a world first for robotic monitoring and treatment of slugs, and like the Blackgrass project, is funded by Innovate UK. The SlugBot project combines hyperspectral imaging expertise from CHAP with machine learning and mechanical engineering expertise from SRC to build an end-to-end system. In this project so far, the team at CHAP have been collecting slug images using both visible light and hyperspectral imaging systems. and CHAP and the Small Robot Company are currently collaborating on training machine learning models for slug detection. We have a research collaboration ongoing at the moment, looking into how we train these models from visible light imagery along with hyperspectral imagery. Ultimately, we want to determine which are the most beneficial parts of the spectrum to use for slug detection so that we can build a practical system for in-field use. Finally, the Small Robot Company are building a precision spraying robot for the actual dosing of the slugs, and then all of these components can be put together to make a complete working demonstration system. This is another great example of a collaboration which brings many different capabilities to the table to develop completely novel technologies. So I think the main message that I'd like to convey in this talk is that the collaboration between CHAP, the Small Robot Company, and other partners unlocks many benefits. The Small Robot Company would not be able to move as quickly as we have done on multiple projects without the expertise and facilities provided by CHAP. The fine phenotyping lab's location at Rothamsted makes possible collaborations with expertise across a really wide range of areas, and we're looking forward to many more fruitful collaborations in the future.
I'd like to thank Tom from Small Robots for his presentation. Sadly, he can't be with us today. So any questions for Tom, please, again, put them in the Q&A box at the bottom of the screen. That would be great. I'd now like to invite Charles from Fotenix to present his presentation. Thank you.
Hello, welcome to Fotenix. At Fotenix, we create vision systems to address the information gap between crop health and decisions made during production. Over the past 3 years, we've enabled farm machinery by empowering operation with data-driven insights on phenotypic traits. A lot of what we do includes translating laboratory research towards in-field monitoring. And today I'm going to give a few examples of the brief studies that we've done to utilize the capabilities at CHAP's Phenotyping Laboratory to facilitate that process. But first, a bit of context. Phenotyping is akin to medicine. It requires skilled specialists to monitor plant status, searching for discrete traits, often using quite manual and repeated measurements, despite many early symptoms not being visible to human vision. I think Tom gave some great examples of this before. And because of this, the sector's long been eager to utilize digital technologies to improve automation and reduce resource required to identify new traits. Unfortunately, there's still a big trade-off in the depth of information available at appropriate cost, and also whether it works in a lab or outdoor environment, which I'll touch on later. In terms of our focus, we focus on embedding capability. We create active spectral camera systems that have 3D capabilities that allow in-depth indicators of plant physiology. These are then embedded onto farm machinery to enable effective and high-performance crop monitoring. The key difference between our technology is a cost-effective approach. It uses LEDs and image sensors, which provide a low noise and high resolution operation, and it removes a lot of the variation seen in other spectral sensors with its 3D recompensation. In terms of our cloud libraries, we have quite a growing and vast library with a range of crops and conditions that have been demonstrated over the years. On average, our measurements half the time to detect changes when compared to existing crop inspection. And we also work with both plant science and machinery sectors to develop both detection monitors and then translate and deploy those outdoors. I'm going to give a brief overview of two quick case studies of how this works. In the soft fruit sector, we focused on fruit grading and disease alerts in tomatoes and strawberries. This work originated within Rothamsted and CHAP, developing the capabilities in labs before being translated out onto large commercial farms in the UK. Where the first elements were, they used our Delta platform, which Tom has alluded to before in the fine phenotyping lab. This records 36 wavelengths across the visible to near infrared spectrum. We often use it at the beginning of proof of concepts. It stores a digital twin of the plant's shape and its spectral signature. We then utilize that and interrogate it for areas that we want to detect, find both spatially and spectrally where the information is, and then we deploy those key areas outside. For this example, we actually took the whole system alongside the likes of Lyat in Lincoln and Saga Robotics, and used it to run alongside their UV treatment service for detecting powdery mildew disease. And this is able to inform us what type of capabilities there are, and also what the variations by taking those models outside, so we can adjust with them. That was then deployed again in a more refined system, which focused on the wavelengths where there was the most variation for the monitoring that was important. Oh, ice creams here, guys. And then assist by putting onto commercial producers like Berry Gardens. We are taking this capability now to other crops and into the glasshouse environment shortly alongside these collaborators. Another example, which was alluded to briefly by Tom from Small Robot today, was the capability of using it on wheat, in which we followed a very similar process. We worked alongside Rothamsted and CHAP to provide baseline models of what the grass weed and the crop look like through different growth stages. We use that to measure when is the appropriate time to both monitor and also distinguish between the various growth stages. We then take that capability and embedded it at the early stages on one of Small Robot's platforms. What that enabled us to do was take that capability that was done in lab in a much more cost-refined and effective process, which can then detect blackgrass and other weeds in an arable crop. And as Tom said, we're looking to scale this and look at how we can look over a wider area and improve capabilities by even looking what's happening within a crop rather than just at invasive weeds. In summary, at Fotenix, we believe in enabling art of agriculture, and we do that by sharing the value across key stakeholders. And we believe that this is crucial to enable the industry to address the sustainability and profitability issues seen in food production. To hear more about what we've done, then please don't hesitate to get in touch with ourselves or our partners at CHAP to utilize our capabilities. Thank you very much.
Thank you, Charles. And mine's a chalk house, by the way. Yeah, so any questions for Charles, please put them in the Q&A box at the bottom of the screen. And finally, we'll pass on to Michael from Videometer.
Good afternoon. My name is Michael Carstensen. I'm CEO of Videometer. Videometer makes spectral imaging sensors for seed and grain analysis, and we are making the Videometer Seed Lab, a fine phenotyping system for CHAP fine phenotyping lab. So the properties we are measuring on seeds are things like purity, damages, contaminants, maturity, treatments, germination, and a lot of other useful traits. Measurements are fast, objective, and versatile, and we work on all types of crops, and this system goes for anything from the size of carrot seeds up to broad beans or corn. So Videometer as a company is placed in Copenhagen, and we are working or have been working on seeds since 2000, and we use a special technique called spectral imaging, which is a merger of two very powerful techniques: spectroscopy for measuring non-contact fast chemistry, and imaging for dealing effectively with heterogeneous material. So combining those means that we can do chemistry on heterogeneous material like seeds. Machine learning AI is the glue that makes these fit together and makes it possible to extract fantastic conclusions from the spectral images. Videometer uses a technique where we do not take one image, but up to 20 different images within half a second with different illumination. So we go with LED strobes from UV up to near infrared, and we can combine reflectance imaging and fluorescence imaging into the same instrument. When we take images of seeds, we typically have the seeds presented on a surface, and we extract the seeds even if they touch. We make a database of seeds that you can see on the lower left, and with each seed there are some primary features that can be computed for all seeds. Those are features quantifying color, spectral fingerprints, shape, texture, size, relative size, contour, whether it's smooth or rough, and a lot of other useful properties of the seeds. From the primary features, we can compute secondary features through a statistical model, what is called a calibration model. And in this case, you see on the left, we have barley with fusarium, and we have a detection of the fusarium spectral fingerprint, marking all fusarium pixels with red, gray moles with black. And that means through this calibration model, where we have known samples with fusarium and corresponding images, we can actually make the machine learning compute a model for these secondary features. It could also be physical purity, class damage, seed health, germination, and so on. So the first video I will show you here is a walkthrough of the seed lab system. You see that the seed lab is a Europallet footprint system, 800 by 1200 millimeters, so rather compact. Inside we have the Videometer lab and the autofeeder, so we feed in seeds on the right side. They are phenotyped in the Videometer lab, and corresponding seeds are found in the picking area on the left. We have a high-speed SCARA robot picking all the seeds based on the phenotype from the Videometer lab, and then placing it according to the class that it receives. In the placing area, we can customize a plate or a tray with plates. In this case, you see 7 MTP plates with 96 wells, and here we have replaced 2 of the wells or plates with petri dishes, or we could replace all of them with petri dishes. That means we have some degrees of freedom in terms of how to place the seeds according to the phenotype. We can also have larger buckets which have similar footprint as the 96 well MTP plates. All of these trays are within the reach of the robot and can be directly integrated. The 2 trays or 2 plates on close to the conveyor are the high-speed positions, since the robot needs to travel a little bit less than for the other plates. The next video I'll show you is some preliminary results from the actual system being constructed right now. And this is with maize as an example. So we are picking maize kernels from the belt. You see we do a little bit of hand feeding, and then the kernels are imaged and classified on the fly. So you see that they get a color to the upper left of each kernel, and that corresponds to the class that you see on the table on the right. So when they are classified, the instructions go to the robot where to pick and where to place each kernel. And in this case, we have 3 positions, and they are for good kernels, for moldy kernels, and for insect damaged kernels. In the top of the cabinet, there is a top view camera that makes sure that we can do validated picking. That means we can see if a seed moves when we take the neighboring seed, and we can also see if the seed in fact disappears after supposedly being picked. So this gives you a further security for proper picking and placing of the seeds. After you have done the analysis of all the seeds, you have here all the classes that you can walk through from the database. So these are the virtual seed classes. And then you can go to the physical seed classes in each petri dish and validate the outcome of the sorting operation. So altogether, we have a fast, objective, and versatile assessment of seed and grain quality using the Videometer lab spectral imaging. We measure a lot of different secondary features from each seed, and we have a full documentation in terms of a database of digital twins of the seeds. We can do automated robot picking and placing of seed and grains based on the phenotype, so that you can use the output trays or plates as germination plates or for further processing. The technology of Videometer is proven by more than 300 scientific papers, where more than 120 are within seed and grain. So thanks for your attention during this presentation.
Thank you very much, Michael. If everybody who can would like to join me on the screen, that would be great. Perfect. So Tom from Small Robots, as I said earlier, can't join us. So any questions for Tom from Small Robots, please add them to the Q&A. There's too many Toms in this webinar, including a robot. I've had quite a few questions come through. And the first one, let me just go back into there. The first one was from Santosh saying, do you test the organic pesticides for efficacy? This is a question from Mumbai. I guess I'll go to you, Tom.
Okay, sure. Not yet, but that's definitely a suitable application for what we can do. And we're especially interested actually because we have another CHAP partner down at CABI who develop biological control agents. So certainly for this type of assay, we'd be looking at, or for this type of application, we could be looking at insect damage. And again, that'd be easy to quantify. There's also potential to actually look at the insect movement and how the control agent is actually affecting that. And it gets over the problem of the insects sort of play dead sometimes. So it can be hard to assess how effective that product is. So I guess the answer is not yet, but I think there's a lot of potential for that.
Thank you. I think this one is for Michael. What is the smallest size seed that can be seed sorted, or the seed sorter can sort? I think someone's having a joke there.
Yeah, so the smallest seed is, of course, determined by the platform that you choose. In the seed lab that we have made here, we have a seed size down to small brassica seeds or carrot seeds, and up to, yeah, I guess, the broad bean or corn seed size. So if you go for orchid seeds or small flower seeds, it will not be feasible. You will need a system which is more adapted to seeds smaller than 1 millimeter of size. So I would say 1 millimeter and above.
And Michael, what sort of speed would it be for this system? Is there a range of speeds, or?
Yeah, so I guess the maximum speed is expressed in terms of picking. So picking speed can be up to 2 picks per seed or per second, excuse me. 1 pick per second would be the realistic one due to the surfaces of the seed and the picking accuracy. So I would say with 1 seed per second, then and if you need to pick out 10% of the seeds, then it would be a sorting of 10 seeds per second. So you don't need to sort of pick out the mainstream of the product, but you typically will pick up some foreign material and other seed types.
Thank you. There's another one here. Is there an increasing demand from ornamental or nursery stock growers in the UK to reference the specialist services that we all provide? So I'm not quite sure who to pass that one over to. That's from Phil Evans.
I would touch on that. We have had interest from the sector. I don't think it has the same sort of labour resource savings as some of the other sort of horticulture areas that we look at, but there are significant, obviously the plants are very valuable, both in ornamental and sort of medicinal areas. So it is an area of interest, and some of our advisors are keen on it, but we don't currently have any commercial deployments in there. I don't know if Tom's had any more interaction than we have.
No, I've got nothing to add to that.
Thank you. Next one, I think this one is for Michael again. What solutions are you developing for automatic virus detection in seeds? Or are there any?
I guess this is more for Tom actually, because I mean we are not a microbial lab or an actual seed lab. We are a developer of a tool. So I think this is an excellent thing for CHAP and for Rothamsted to look into. We have different papers that have looked into viruses and viroids and bacteria and fungi and so on. So I will leave that up to the experts, but there are some results on this, and I think Tom may know more of that.
Yeah, I'm not sure what work's been done there, but I think it's certainly a really interesting question, because obviously virus transmission in seeds is a really important issue. So I think that is actually something that would be very interesting to look at and see whether we can monitor that and detect the contamination. I think a lot would depend on actually whether or not that was showing up actually in the seed coat, or whether it was purely in something that you couldn't detect and internal. I think until we try it, it's hard to say.
Thank you. One for Charles here again. Are there any spectral imaging systems that have been tested in drones for wider crop applications?
So there are quite a few spectral imaging systems that have been used on drones. Most of them don't have the active method, so they use filters rather than ours. So we don't typically work with drone applications. And there's some great uses of them. I think where I would heed caution is that spectral imaging is very variable. Plants exhibit, for technical people, non-Lambertian reflectance. So basically means you look at them from different angles, they appear a different color. And unless you can record and remove that variation, you're quite limited of what insight you can take from that. So I think there's definitely a lot to be done in the handover between ground-based and drone-based systems, because each has significant benefits in slightly different areas versus coverage versus sort of information and detail.
Thank you. Tom, I think this one's for you. Is the performance of reflectance imaging equal to that of multispectral imaging? And if so, can qualitative data be expected from low-cost reflectance imaging for early disease detection in vertical farming, not phenotyping?
Well, I mean, I guess so. Multispectral imaging is based on reflectance. But in the case of sort of multispectral as compared to RGB, obviously with RGB, you're just looking at the 3 bands within the visual range. With multispectral, you're looking at additional bands, but in both cases, you're just looking at reflectance. So I guess the simple answer to your question is yes, obviously reflectance is really important, or is really useful for detecting early disease, particularly when it's used sort of in the context of multispectral imaging.
Thank you. This one, can blind plantlets be detected within a 200 cell flat, and in brackets, tomato seedlings? Is that one for you, Michael?
Yeah, I'm not sure.
I'll have to maybe get back to you on that one.
So I think that is something we should look into before we shoot from the hips.
Thank you. I've got a few more questions here. One for Charles here, actually. Why has digital technology seen a lack of adoption in phenotyping?
That's quite an interesting one, and I think it's a combination. I think it's a combination of where the sort of specialties sat within those doing the sort of phenotyping at scale, and also how scalable the opportunities were. We've seen a lot of digital technology in terms of color imaging and phone apps being utilized, because they're quite a good step in between the sort of manual recording of it. I think it's going in the way of the medical sector in terms of assisted diagnosis. But I think there is that model, that trade-off of whether plant breeders are going to have to become data scientists or whether there's the right model to do it. So in short, I think there's been a bit of a gap between what was possible at the right price point for that kind of imaging. Not obviously a lot of breeding can happen in high-end labs, but a lot of it happens out in the field. And if you can't do the same analysis, then it's really difficult to do the full sort of cycle. But the right companies are looking at it. So I think there's some significant movements in that field, and we look with quite sort of excitement of what's to come in. I think they call it sort of digital agriculture or digital phenotyping, but you have to keep up with the names these days.
Thank you. There's one here for you, Tom, from Catherine. Are there greater benefits or negatives from imaging for efficacy testing in the lab environment, e.g. no environmental impacts?
Yes. So, I mean, I think the advantage of lab-based phenotyping is, as you're suggesting, is that you can control all the variables. So it's really good if you want to look very carefully at, say, different formulations, and you want to be able to just, compare a small number of formulations under very carefully controlled conditions. But then the disadvantage is that you're sort of limited by scale. So again, if you wanted to maybe look at many more treatments or more on the sort of glass house scale, then that's the disadvantage of doing it in the lab. So yes, I think it depends on the application, but certainly if you want to very carefully assess a product, I think lab-based phenotyping has got a lot of advantages.
And then one more for you here, Tom, is what type of samples can you image in the PhenoCenter? I mean, how big can the plants actually be?
Right, so I guess there's two types of samples that can be conveniently imaged. First will be samples in multi-well plates. So for example, they might be like leaf discs. For example, you're doing like an insect feeding assay. Maybe if you're looking at insect movement, how biopesticides affecting that. Or even if you're looking at, say, looking at fungal growth and doing competition assays, that type of sample. You can also then look at small plants up to about 20 centimeters in size. So I guess that's sort of type of samples.
Thank you very much. And I think then maybe the final question for maybe Michael, depending on how long it takes to answer. Will you be able to make randomized samples of good crop seeds and place them on a germination paper in your new bit of kit?
Yes, so in the placement area, you can place anything up to the size of the sort of exterior boundary of the robot reach. So definitely you can place a germination paper and put all the seeds in correct positions on the paper. So that would be an obvious application.
Great, thank you very much. Does anybody else want to say anything in particular before we close? Got any points you want to raise from the panel? No, that's great. Well, thank everybody for coming, and thank you very much to the presenters. If I can ask them to turn their screens off now, that'll be great. I'd just like to thank everybody who has come along to the webinar. And obviously, if you would like to ask any questions, please take a photograph of my email address and contact me. So if you're interested in any projects, either grant funded or commercially funded, or you're interested in membership, or you're interested in just receiving our newsletter, any queries whatsoever, please do not hesitate to contact me. And I hope you enjoyed the webinar. Thank you very much.